All right, I think it's time to start. Thank you everyone and welcome to our Nantucket Field Station Spring Webinar Series. My name is Yvonne Valancourt and I'm the director of the Nantucket Field Station. We are a facility of UMass Boston. We are managed by the School for the Environment, and we offer courses and support researchers, not just from the UMass system, but from other agencies and institutions. We sit on over 100 acres owned by the Nantucket Conservation Foundation. We are joined tonight by two speakers who I will introduce shortly. I would first like to say a word about the format of this webinar. We will have two short talks, or maybe not so short. And if you have questions as you listen to each speaker, please use the question and answer chat box. Type your question in there and include the person's name who you're posing the question to. The sooner you put the question in, we'll be able to ask directly after the speaker finishes their talk. And that question will be facilitated by Leo. Leo Stella is a student from UMass Boston from the Department of Urban Planning and Community Development. And he is assisting us with these webinars, and for which we are very thankful. So without any further ado, we are being joined by two people who both are part of the were part of the Jarrett Byrnes lab. So our first speaker this evening is Marc Hensel, and though he is currently a postdoc at the Virginia Institute of Marine Science, he was part of UMass Boston where he completed his PhD. And people from Nantucket, you may recognize his face because he spent a significant amount of time here on Nantucket and Folgers Marsh. And rather than listen to me keep going on, I will turn the floor over to Marc. So please welcome Marc Hensel. Thank you.
Thanks, Yvonne. Let's get this all set up. All right, we looking good? Cool. Okay, so thank you very much for that introduction. I'm really excited to tell everybody about this research today. So this is some research I did during my dissertation at UMass, like Yvonne said, where a big chunk of this was done out on Nantucket. And oops, I clicked the wrong thing. And basically, this is a part of my dissertation that was seeking to understand and describe the wide-reaching effects of predators in New England salt marshes. So ecosystems are fundamentally different with and without predators. The loss of predator species can have impacts that cascade down a food chain to plants and altering basic ecosystem processes. A famous example of this is the kelp, sea urchin, sea otter food chain in the Northeast Pacific, where the hunting of otters by fur traders caused a huge increase in their sea urchin prey, which overgrazed kelp forests. This led to the loss of a number of other species that depend on kelp, and importantly had knock-on effects to the coast with increased coastal erosion and storm damage because kelp is a primary buffer from wave action. This phenomenon is called a trophic cascade, and it has been found in many ecosystems. Since this research came out, that we've really started to understand that predators actually affect more than just primary production, because predators have wide-reaching roles in natural ecosystems through effects on their prey. So by both consumptive effects, eating, and non-consumptive effects, scaring, predators can control a lot of different ecosystem processes. Predators can control a whole food web structure, not just their prey, but other species as well. The amount of predators in a system can determine energy flow, nutrient cycling as well, and even the structure of habitat in the case of ecosystem engineering predators. And so out in nature, how predators affect their prey is totally determined by what that predator community looks like. So the broad role, the strength and scope of predator effects really depends on Basically the group of interacting predator species in a given area. An awesome example of this is in New England salt marshes, where the trophic control of a sap-sucking insect is totally dependent on basically which spider predator species are there. Some spiders only eat the plant hopper, where other spiders eat each other and the plant hopper. And so plant growth in these areas is totally dependent on predator diversity, the number of species, and predator identity, which one is present. So these key components of predator community structure really need to be understood to figure out basically how predators affect whole ecosystems and where predator conservation is going to be needed most. The way that I approach my research on this subject is sort of like this here in this diagram, where predator communities control prey communities, and the prey communities do different things. There's grazers, decomposers, filter feeders, burrowers that all affect different ecosystem processes, or we can sometimes call those ecosystem functions. And then, of course, overall ecosystem processes are affected. And so we know a lot about these arrows that I've drawn, but we do not know a whole lot about this arrow here, the indirect effect of predator community structure on overall ecosystem processes, because this is a difficult question to tackle and represents a whole new area of ecology. Luckily, salt marshes are an awesome place to ask questions about the role of predators in overall ecosystem processes. In New England in particular, salt marshes of New England have all these resident invertebrates, crabs that are burrowers or herbivores, filter feeding mussels, snails that decompose dead tissue, that all control and help control many important ecosystem processes. These invertebrates are all hunted by a really cool, diverse, abundant group of cross-ecosystem predators. So as the tide goes out and low tide is exposed to the creek bank, birds come in and feed on the marsh edge. This picture is taken by Lenny, the edge of Folgers Marsh. And then when the tide comes up, from the marine side of the food web, species like striped bass, eels, blue crabs, green crabs, mummy chugs come into the marsh and take advantage of all these resources too, right? And so this is the New England salt marsh predator community, and the way that I've structured my research questions kind of go along with this picture right here, basically where there's a marsh predator community, birds, fish. This marsh predator this marsh predator community has different guilds, and from marsh to marsh, they vary in abundance, in diversity, in the number of species. Right, these predators control these predators feed on prey that are like decomposers, herbivores, burrowing crabs, filter feeders, which then in turn, of course, affect the different ecosystem functions like primary productivity, decomposition rates, sedimentation rates, the way in which sediment is deposited onto the marsh. So we know a lot about these arrows right here, how predators affect their prey, how prey affects ecosystem functioning. And my research is then, that I'm going to talk about here, I used experiments and surveys to sort of understand this dotted arrow as well. Really quickly, I just want to define one term that I'm going to use just a little bit, which is multifunctionality. This is the simultaneous performance of multiple ecosystem functions. So these different ecosystem functions here, basically what multifunctionality is just a way to summarize the overall combined rate of a bunch of different processes at once. So it helps us look at overall process. So to address this, how do predator community structure affect overall marsh processes? I have two major research questions that I mentioned. I used experiments and large-scale surveys to understand or to answer, excuse me. So first, how do bird and marine predator guilds affect marsh invertebrates and overall ecosystem processes? So to answer this question, I did a manipulative field experiment in Folgers Marsh, where I manipulated the presence or absence of these predator guilds, and I measured the rest of this diagram. I measured the community structure, so the resident invertebrates that live there, and these ecosystem processes. how I manipulated these predators, in Folgers Marsh along the creek banks, I had these 2x2 meter plots where I either excluded birds. That's what this picture is on the right. It's tough to see, which is the point. This is a fishing line wrapped around PVC poles. And sometimes I use some flashing tape as well as a visual deterrent to exclude birds. And then to exclude marine predators, I built this cage here, which is a floating net cage. I've got a little video that hopefully plays okay. Where as the tide comes in, the water raises the floats, and no fish or crabs or anything can swim into this. And I have treatments where both are excluded and neither are excluded. So over the course of the growing season, I measured invertebrates and ecosystem function. So what did we see? So at the end of the experiment, I found that birds and marine predators have different effects on the marsh invertebrates. So I'm going to show the graph on the left, but it's a little bit complicated. It's much easier to just look at my pictures on the right. But what I found was that marine predators, so the crabs and the fish, control all the different prey species that I measured. But bird predators, in fact, only had an effect on crabs, on the burrowing fiddler crab and the herbivorous purple marsh crab. So importantly, bird and marine predators do different things. Sometimes they complement each other, and sometimes they don't. How does this translate then to ecosystem function? So here on this graph, this is the rate of overall ecosystem functioning, where higher numbers mean the more processes that are occurring at higher rates. And my different treatments down here, which you don't need to worry too much about, what I found was that the plots that had the highest amount of functioning were the plots where all predators were allowed in. And in fact, the lowest combined rate of ecosystem function occurred where no predators were present. So bird and marine predators together enhance ecosystem multifunctionality in my experiment through these additive effects on their prey, right? So this was a really interesting result, and I think this told us a lot about what happens along the creek banks in plots in a manipulative experiment. But I also wanted to understand basically how does this scale up to the whole marsh, and how does this scale up to places that aren't Folgers Marsh as well. And so to get at that, question number 2, what are the marsh scale patterns and correlations between predator communities, the marsh invertebrates, and ecosystem function across New England, or across Massachusetts, really? This was a MIT Sea Grant funded project I started right when I got up to Massachusetts and working with Jarrett. And basically what we did, so these are our sites on the left here, is we went to all these marshes and chunked them up into these 3 different zones, and we went out there and quantified, everything you're looking at here, all of the predators, all of the invertebrates, and all these ecosystem functions. And then we looked at relationships between the predator community. So marsh predator diversity, how many different species, the abundance of all predators, and then also within the guilds, bird and marine. We measured again marsh invertebrates and quantified all these different ecosystem functions. And so I'm going to walk through basically each of these black arrows and the direction of each of these. But really quickly first, since this, for the context of this talk, I do want to point out the patterns in bird and marine predator diversity across Massachusetts. So on the left, this is birds. On the right is marine species. Each of these circles is one of the sites. The size of the circle is abundance, and how red it is richness. We found 65 total bird species across our surveys, 22 total marine species. And I in particular wanted to point out how well performing the Nantucket marshes were. So the 3 marshes that we surveyed here were Folgers, Medowee, and Eel Point. And basically Nantucket marshes in our surveys were the most diverse and most abundant fish and marine predators throughout the places that we surveyed. 36 total bird species. I think it was that was in Medowee Marsh, 16 total marine species in Folgers Marsh. So this is actually why I decided to do the experiment where it was, but it was kind of cool that Nantucket had such good, abundant, diverse fish and predator communities. So what does that mean though? Not all marshes have all of the birds and all of the fish, right? And so to understand that variation, right, we're gonna look at this diagram here. So first, the relationship between marsh invertebrates and multifunctionality. This is actually something that's been teased out in experiments before. Marsh inverts have a positive effect on multifunctionality, and this was confirmed again in our surveys, where mussel density, snail density, fiddler crabs, they all increase the rate of multiple processes happening at once. So what about the effect of predator community structure, diversity and abundance, bird and marine, on the resident marsh invertebrates? So as you can guess, there's a lot of different predators and a lot of different invertebrates. So I'm not going to show a bunch of graphs. I'm just going to plop up this table here and show that predator diversity and abundance both affect invertebrates in different ways. Abundance generally decreases, predator abundance generally decreases invertebrate abundance, but basically there's complementary effects of both species richness in predators and abundance. And when we look at birds and marine predators, we see a similar pattern where there's an interaction between richness and abundance, both within marine and bird predators, correlating with resident invertebrate density. So again, similar to what we saw in the experiment, bird and marine predators and predator diversity and abundance do have different strong effects on their invertebrate prey. So now what about this last connection here? How does predator community structure affect multiple ecosystem functions? So I'm going to show a couple of different graphs here, but basically the main point is that total predator richness and abundance together interact to increase the rate of ecosystem function. And it's really cool because they kind of compensate for each other. So on the left here, I have two plots. This is marshes that have low predator richness, and this is marshes that had high predator richness. And so increasing predator abundance has a stronger positive effect on multifunctionality at low richness. And then the flip side of that is also true, which is really cool. That means that predator richness, increasing the number of species, has a stronger positive effect on multifunctionality at low abundances. So again, there's this really interesting compensation between abundance and diversity, and it makes sense that the most abundant and diverse marshes would be the most multifunctional. Within the guilds, we found that not birds, but actually marine predator richness did increase ecosystem functioning as well across all of our surveys. So this is what our sort of final diagram looks like here, with this indirect positive effect of predator communities on multifunctionality. This is a really neat finding because those components, those different components of the predator community have different effects on the marsh invertebrates, and that combines to increase multifunctionality, both on small experimental scales and on large, whole marsh scales. And so what this means is, to determine this link between predators and large scale processes, we have to look more broadly, across nutrient regimes, under different levels of climate change. And so like the final sort of diagram looks something like this, where environmental and abiotic factors sort of affect everything. Predator diversity and abundance and identity are all important. And multifunctionality is related to ecosystem resilience, where an ecosystem that's performing more functions at higher rates is going to be more resilient over time. So from a conservation standpoint, there's a couple of big take-homes that I just want to point out. One is that in Northwest Atlantic marshes, fragmentation, overharvest, habitat destruction are big threats to marshes as a whole. These are also things that will affect predators. So that means that there's the potential of amplified effects on both from bottom-up standpoint and top-down forces in marshes if these things continue to go unchecked. And also, when it comes to the conservation standpoint, especially with predators, we have to focus on conserving, the whole community structure, not just its components like diversity, because that's what becomes really relevant across large spatial and long temporal scales. So with that, I'd like to thank a lot of different people. So a ton of this research was funded through the Nantucket Conservation Foundation. Obviously, a big chunk of it also took place, on Nantucket as well. So, big thanks to them and to the NBI as well. And to my advisor, Dr. Byrnes. This was a project that we started right at the beginning of my dissertation and carried all the way through, and it's still going now. And so I'm really excited for the future of this work. Had a number of really good undergraduate researchers help me out throughout the course of this project. And with that, I'll turn it over to Leo to ask any questions.
Awesome. Thank you, Marc. People are maybe being a little shy. We don't have any yet in the question and answer portion. Just so folks know, there's a chat feature and a Q&A feature. And I don't believe that I can, oh, look at that. Yeah, I can allow John to talk here. So I'm gonna allow John to ask his question.
I think that when I think of ecosystems, I think of a basic equation, A plus B equals E. You focused mainly on the biota, and yet you mentioned spatial and temporal issues. Did you do anything regarding the abiotic factors, or did you just concentrate on the biotic factors?
Great question, John. And you know, it's really, if you sort of scale up the diagram that I have of, you know, how I envision everything working at the end, you know, that's where you know, different abiotic and environmental changes can potentially come into play. So, you know, I did a number of things to control for differences in abiotic forces and environmental forces. For example, even when it comes to the food web surveys itself, you know, we try to do those in the same time of year, you know, to catch, so you know, we're not getting migratory birds at different times of the year and stuff like that. And then, you know, we try to select marshes of similar size with similar creek size, which has a big effect on large-scale processes like sedimentation rates, that sort of thing. So, you know, we did do as much as we can to make sure that those differences in abiotic forces are the same in a lot of different marshes. But as for, like, you know, sort of explicitly working that into to our models and stuff, that was not something we were able to do on for this project. But I mean, that's sort of an overarching goal in ecology in general, is to be able to basically handle it all. And you know, that's not something we did with this project, but definitely something that's on our mind. So yeah, good question.
Familiar with Plum Island and Belle Isle, and also Folgers, and they are different, and that occasioned my question.
We've got another question here in the chat, so I'll just read it out. The question here is, when Bernstein excluded blue crabs from Chesapeake Bay benthos, infaunal abundance increased dramatically, and so too did benthic infaunal species richness. Do New England marsh benthic infauna behave similarly when crabs are excluded? That's from Eugene Gallagher.
That's a really cool question, Eugene. The short answer is we don't know. This is definitely something that is on the mind of researchers. It might even be on Michael's mind, because as blue crabs move, you know, further up north, this is going to be something that that needs to be studied. So, you know, I'm not sure of maybe the current status on this, but when I was like doing this, these projects, Nantucket was one of the only sites that I went to that even got blue crabs. I think maybe we got some on the Cape as well. But definitely not, I don't remember seeing very many north of the Cape. I think that maybe has changed over time, and maybe Michael and I can talk about this in the panel potentially. But the direct answer is we don't know yet, but very likely, yeah, blue crab impacts, as because they're, I mean, probably, you know, they're an extremely important, if not the most important predator as you go up the coast. That's gonna be something that's really interesting for the future of New England marshes.
Awesome. Thanks, Marc. And we'll just do one more question before we turn it over to Mike for a presentation. Just a reminder for folks, there will be time for more questions later. So Heather's asking, did you observe overpopulation of other species when you excluded predators?
Oh, good question, Heather. You know, we didn't observe any like overpopulation, you know, big booms, really, but there were a couple of interesting findings sort of related to that. So in Folgers Marsh, in just sort of like as you walk along the marsh, no exclusions or nothing, you at least when I was working there, you very rarely see any snails, littoria. But as soon as I started to exclude marine predators, I noticed that these snails were recruiting into those plots. So this was a really interesting finding for me because this meant that basically as soon as the snails were recruiting to these areas, but they get eaten up like right away, either by little crabs or by mummy chugs who just take a mouthful of snail very, very easily. So you know, I think potentially if things were to go like over time or multiple seasons, there potentially could be a buildup of snails, maybe even of purple marsh crabs as well. But I didn't observe any of that in my experiment.
Thanks, Mark. This is so interesting. We could keep asking you questions, but we're going to pause that. So for those of you who have more questions, write them down. But we're going to now hear from Michael Roy, and it's my pleasure to introduce him. Normally we would be seeing Michael down here on Nantucket, but tell us about your research.
Thank you so much for that introduction, Yvonne, and excellent talk, Mark. I wanted to mention that on the blue crab front, we actually, so I've done a fair amount of work in Plum Island, which I'm going to talk about a little bit today as well. But we saw 5 blue crabs up in Plum Island this summer, which was really wild to see. So they're coming up, and it'll be interesting to see how, you know, their presence is really going to impact, you know, food webs, you know, up in the pie. So. All right, so I'm going to go ahead and get started on sharing my screen. All right, thank you so much, everyone, for coming to our talks. I just want to start by highlighting my 2 collaborators, Dr. David Samuel Johnson, who is also at the Virginia Institute of Marine Science. He started and pioneered a lot of the work that I'm working on currently. He is the first person to find the fiddler crab that I'm going to be talking about in Plum Island. And of course, I want to talk about my advisor, Dr. Jarrett Byrnes, who's advising this project would not be possible. And I also want to bring up first and foremost my funding sources and acknowledgements, a whole series of people that have really helped with this research, a number of undergraduates, research funding through NSF, the Battle Fund at UMass Boston, UMass Boston itself, and some internal grants at UMass Boston. So with that, I'm going to get started on talking about really the work that I started in 2017 and sort of have worked on throughout my dissertation. And it's really sort of an ecological story that I'm going to be talking about. With any ecological story, with any story, I want to set the scene. So first, I'm going to talk about the star of the show. The fiddler crab, Minuca pugnax. This is the mud fiddler crab is a burrowing species of crab that is native to Nantucket all the way down to Florida and is a really critical species. And the 2 places that the 2 scenes, the 2 locations that I'm going to be talking about are the UMass Boston Nantucket Field Station, specifically Folger's Marsh at the UMass Boston Field Station, and the Plum Island Estuary, specifically sites at the long-term ecological research site there. Okay, so let me introduce you to the star of the show, the mud fiddler crab, Minuca pugnax. So the image on the left, you'll notice it's a very lush green spot. This is the low marsh of a typical salt marsh. This is where the tall Spartina alterniflora grows. It's right next to creeks, and part of the reason why it grows so tall is because it's the first grass that's going to encounter all of the nutrients that come in on every tide. And so this is the spot where the fiddler crab, Minuca pugnax, lives. And where does it live? Well, it burrows into the sediment around the Spartina alterniflora. So it digs these deep holes, roughly 15 to 20 centimeters in depth. And it's a really dynamic species because it potentially has ramifications for interacting with soil microbial activity and water saturation, because it's basically Swiss cheesing the marsh. You'll notice here that the male fiddler crab is front and center. He is characterized by that large claw that looks like he's holding a violin, like a fiddle. And then a female fiddler crab is to the right of that in this image. And so these burrows are really dependent on the kinds of sediment that they're burrowing into. not all salt marshes are created equal. Some salt marshes are going to be clay rich, so more peaty. Some are going to be a little bit sort of a mix of the two that are sandy, and some are going to be more of like sand flats. And this image from Chen et al. Highlights fiddler crab species that are not the mud fiddler crab, but they behave similarly in terms of their burrowing behavior. And so you'll notice that the clay-rich mudflat, fiddler crabs are able to burrow deeper, they're able to have wider burrows. And part of the reason for that is because the clay-rich mudflat has a little bit more of structural integrity to maintain those burrows. And fiddler crabs don't necessarily have to spend as much energy maintaining those burrows once they're made. And then as you'll notice that as you go towards sand flats, they're a little bit shallower and then not quite as wide. And so this is really important to take into consideration for the rest of the talk, as well as just thinking about how fiddler crabs interact with their environment. And what's really important to understand is that fiddler crab densities can get pretty high. So they're really creating a lot of burrows into the sediment. They can get upwards of 150 individuals in some southern salt marshes in Nantucket, between 80 and 100, and then even more. And so they are really critical species for interacting with this environment. So let me talk about the Folgers Marsh and the University of Massachusetts Nantucket Field Station, where this work was done. So as Marc mentioned, it's an incredible place to do research. This is looking out over the bluff with the bunkhouse behind me when I took this picture. It's really just a stunning place to do research. And there are a whole suite of species that exist that live there, as described by Marc during his talk. So as I mentioned, Spartina alterniflora is the species of grass where fiddler crabs occur, but in southern salt marshes, what I mean by that is salt marshes south of Cape Cod, they're predominantly dominated by this species of grass. North of Cape Cod, they're dominated by another species of grass, Spartina patens, which I'm going to talk about on the next slide. But it's just critical to keep that in mind in terms of differences between different sites. The last thing that I want to talk about for Folgers Marsh is that there are a lot of oceanic inputs of sediment. So every tide, sediment gets flushed into the marsh, and that's and by and large how the marsh builds up both vertically as well as horizontally. And so it's really sort of how the marsh grows or maintains itself over time. And because it's oceanic, there's a lot of silt, there's a lot of sand, so it's a little bit more of a sandy and silty salt marsh. So this is where fiddler crabs live. That's their historic range. Fiddler crabs, as I mentioned, have a range all the way down to Florida. But what was really interesting is that in 2003, Marc Sanford et al. Found individuals of the mud fiddler crab for the first time ever in their life history in Scituate, Massachusetts in 2003. Then, as I mentioned, David Johnson and other colleagues at the Pi LTER, but primarily David, found individuals in 2014 up in Pi. And so this is the first ever occurrence in northern New England salt marshes. They're now found as far north as southern Maine. So their distribution is really expanding. So the question is, why is their distribution expanding? Well, a big piece of that is likely due to the fact that the Gulf of Maine is warming at a rate that's faster than 99% of the global ocean. So you can really see here that the Gulf of Maine is highlighted in sort of the red colors here, which is the more extreme habitats that are experiencing warming. Part of the reason for that is because due to the oceanic offshore shelves, as well as other morphological features, the Gulf of Maine sort of acts as this large bathtub where water recirculates. It's still colder than marshes and habitats south of Cape Cod, but it's much warmer than it used to be, which is the theorized mechanism for how this fiddler crab was able to establish itself. So now let me go into the second location, the Plum Island Estuary. So it's a really gorgeous site as well. It's an extensive salt marsh. It's an enormous salt marsh. It's actually it's the largest continuous salt marsh in the Northeast. So it's dominated by this species of grass, Spartina patens. It does have Spartina alterniflora, but in less abundances. And this marsh really extends, all the way from New Hampshire to Gloucester. It's really a large dynamic system. You'll notice that Plum Island itself is this sandbar island towards the ocean. All of the marshes are to the west of that sandbar, and it's really a critical habitat. What I want to highlight before sort of going into some of the research that I've done is the fact that this is a peat salt marsh. So it's really sort of like a dense peat organic matter, and it has more freshwater riverine inputs of sediment. And so that really sort of changes the sort of, it's quite different than the Nantucket field station Folgers Marsh from that perspective. So in terms of fiddler crabs interacting with their environment based on this burrowing behavior, what we know is that fiddler crabs mediate ecosystem functioning, which is that the functions that are occurring in salt marshes or any ecosystem, as Marc mentioned, differently in different locations. So on the x-axis, we have different locations here. On the y-axis, we have soil strength, which is just a measurement of soil compaction. You don't have to worry about what the different colored bars indicate, but what's really important here is that all of these bars and all of these sites, this is the study done by Williams and Johnson, that really, it was a study looking at the effects of fiddler crabs and the purple marsh crab Sassarma on different ecosystems. And what this highlights is that fiddler crabs are not uniform across different sites. And a large part of that is due to the fact that sites are different between locations. With that being the case, or site level ecological characteristics are different between different locations. So that being the case, it's going to be difficult to predict how fiddler crabs will interact in their new sort of expanded habitat than potentially how they're interacting in Nantucket. So that leads me to my two research questions, which is, to what extent does this fiddler crab impact ecosystem functioning? And the three ecosystem functions that we looked at for this study were soil strength, primary production, and decomposition. In its historic range, which is Nantucket, versus its expanded range in Plum Island. And then the second question is, does the density of fiddler crabs influence any of these impacts that the fiddler crab has on its on these ecosystems? So to answer these questions, we set up a series of fiddler crab inclusion cages. And so we put in different densities of fiddler crabs, ranging from no crabs, which is demonstrated by the box all the way to the left, which is 0. Then there is 20 crabs, which demonstrates ambient density. And this is crabs per quarter square meter, which is about ambient in Nantucket. So we replicated this 4 times in both locations. So 24 total cages per site. We ran this experiment from June to September in 2017 and 2018. We measured a whole different suite of environmental indicators for these ecosystem functions. I'm not going to go through this laundry list, but what I do want to highlight is the metric at the bottom, which is decomposition. In order to measure decomposition, we took Spartina alterniflora, we dried it, we put it in these mesh bags, and then we buried it 5 centimeters below the surface of the soil and 15 centimeters below the surface of the soil. And the reason for that is because we wanted to determine decomposition rates at these 2 different depths, because depth makes a big difference in terms of decomposition rates. And so before going into sort of how we took all of this environmental data and really tried to derive inference for how fiddler crabs were really structuring these ecosystems, how they were really sort of driving ecosystem functioning, I want to go into sort of like the what these cages looked like. So it was pretty straightforward. It was just 4 PVC pipes with mesh around them buried into the sediment to prevent crabs from burrowing underneath. And then we put steel flashing at the top to prevent crabs from either crawling out or for new crabs to crawl in to really control for that density. So as I mentioned, we have all of these environmental indicators, soil strength, primary production, biomass of Spartina alterniflora. And so given the fact that we have all of these different environmental indicators, we really wanted to tease apart what's really driving the system. And one methodology that is used quite often is using causal models and structural equation modeling. So I'm going to walk you through this methodology to really sort of understand how these species are interacting with this ecosystem, how this framework works. So it's very straightforward. So let's say you have variable A and you have variable B, and variable A causes some change in variable B. And that change is demonstrated by an arrow going from A to B. You can also have another variable or another species. So A causing a change in B causing a change in C. And then you can also have A causing a change in C as well. So it really sort of qualitatively gets the researcher understanding about how their system is working, how they believe their system is working. And to test this quantitatively, you can use what's called structural equation modeling, or SEM. And structural equation modeling takes this causal model and it applies statistics to it to get statistical outputs to determine how likely this causal model represents the world, represents reality, and it gives you statistical outputs to determine, if it is representing reality, what's actually driving, what variables are driving change? What is the causal relationship here? Okay, so with that in the back of your mind, I'm going to jump into my results. The results I'm going to show you are a mix between both the structural equation models that we developed, as well as some heat maps that show some of the interactions that we found. And so on the left, you'll see that we have crab density, which is just the number of crabs per plot, and that we manipulated. Those are the crabs that we put into each cage. And then we have initial soil strength, which is just the soil strength that we measured at the beginning of the experiment without manipulating anything. And this was measured within each plot. And so then we wanted to determine, does crab density or initial soil strength affect the number of burrows at the end of the experiment? And this is important because it is that burrowing behavior that's going to be driving whether or not the fiddler crab is going to be interacting with the ecosystem in some way, in either Plum Island or in Nantucket. And so what we found is that the thin gray arrows indicate a non-significant relationship, that crab density by itself and initial soil strength by itself does not cause a change in burrow density. It doesn't matter how many crabs you have, burrows are irrespective of that, except when taking into consideration the interaction between the two. So essentially, as you increase the number of crabs, you also have to increase the amount of the strength of the soil in order to increase the number of burrows. And so it goes back to that figure that I showed before, which is that that peaty marsh is going to really be facilitating the ability for crabs to maintain these burrows and to increase burrow density over time. So this is all well and good, but this doesn't really talk about the actual ecosystem function, which is soil strength. So does burrow density actually confer something about soil strength? And the answer is yes. So the thick blue line indicates that there's a significant causal relationship between burrow density and final soil strength, such that as you increase the number of burrows, you are decreasing the structural integrity of the soil. You're decreasing that compaction, which sort of from a first principles perspective isn't that big of a stretch, because when you think about all of that water saturating the sediment, you're sort of increasing the amount of water between sediment grains. So it makes sense that might be the case. This final gray line just indicates that initial soil strength has a causal relationship with final soil strength, which is what we expected. So this crab density soil strength interaction can be a little bit difficult to visualize. It was difficult for me to visualize at first. And so what we did was we plotted this in a heat map to really sort of try to understand how these three different variables are interacting with one another. So on the x-axis, down here, we have initial crab density, which is crabs per plot. So again, that's just that metric of the crabs that we put into each cage. On the y-axis is this initial soil strength, which is, again, just that soil strength that we measured at the beginning of the experiment. And the different colors represent the number of burrows. So the redder the color, the warmer the color, the more burrows you have, and the cooler the color, the fewer burrows you have. And so this really highlights that in this upper right-hand corner, as you increase the number of crabs and as you increase soil strength, you have more burrows. And so this really highlights that interactive relationship and how fiddler crabs are really or how these environmental properties and population properties are really driving fiddler crab burrowing behavior. Okay, so what about the next ecosystem function, primary production? So I just want to mention that I threw sort of this model up. This is exactly the same model that I showed before. The thing that I want to highlight, though, is that what's really interesting is that fiddler crabs may be self-sustaining in terms of their habitat. That as you increase the number of burrows, you're decreasing soil strength, which is going to decrease the capacity for fiddler crabs to maintain their burrows. So it may actually be facilitating fiddler crabs in sustaining their own habitat, because there may be sort of this this pulse where you have really strong sediment, so a lot of burrows, but then maybe the next year you have weaker sediment, and then so on and so forth. So I really sort of just want to highlight those dynamics before talking about primary production. So we measured primary production as both the amount of shoots in each plot, as well as the biomass of the grass that we basically cut, dried, and then weighed at the end of the experiment. So the Spartina biomass was not affected directly by any of the metrics from fiddler crab activity, basically indicating from this first approximation that fiddler crabs do not affect the amount of Spartina alterniflora grass where they occur. Except again, when talking about it from the perspective of an interaction, we found that there was an interaction between the soil strength when we collected this grass, as well as the shoot density when we collected this grass to weigh and dry. And so there is this indirect relationship where if fiddler crabs are affecting soil strength will have an effect on Spartina biomass. So what does this interaction look like? Well, so there is we created another heat map that demonstrates this interaction. So on the x-axis is final shoot density, which is just shoots per quadrat, final soil strength, which is again measured in PSI. And this time, the warmer colors indicate more Spartina biomass, and the cooler colors indicate less Spartina biomass. And what you'll notice here is that as you increase the amount of shoots and you decrease soil strength, you actually increase the amount of total biomass of the Spartina alterniflora, which basically indicates that Spartina likes to grow a lot of shoots, but it doesn't like super, super tough sediment. And you can imagine that being the case because part of the way that Spartina grows is creating rhizomes under the sediment and then shooting clones through the sediment. And if it's really hard to grow, those rhizomes, it's going to be hard for the plant to grow shoots. And so in this instance, fiddler crabs may be an indirect positive benefit to Spartina biomass via its weakening of the soil, at least at different time scales. Okay, so the final ecosystem function that we looked at was decomposition. And again, so this is more or less the same plot as before. I will highlight that this is only 1 year of data. It was only in 2018. We tried to collect this data in 2017. The methodology that we used failed. We used tea bags. They disintegrated at the end of the experiment. We used these litter bags instead, these mesh bags, and that worked great. And so we do have data for this. So some of the causal relationships are going to be slightly skewed for the crab density, but we feel fairly confident in the relationships with decomposition based on our statistical outputs. And I can talk about that at the end of the talk if you'd like. And so what we have here is depth, initial soil strength, and burrow density. And then we have this burrow density and depth interaction factor because we wanted to determine, does depth and the number of burrows have an effect on decomposition? And this final litter bag mass is that metric of decomposition. And what this shows is that depth and burrow density by themselves do not affect the decomposition rate. But when burrow density and depth are combined, that is what's driving this final litter bag mass, this decomposition. And so again, I want to highlight this in a heat map to show this interaction. So on the x-axis, we have burrow density. On the y-axis, we have depth, and the scale is inverted such that 5 is at the top and 15 is at the bottom. We created it that way in order to sort of have it like, 5 is shallow and 15 is deep. And so warm colors, again, are an increase in decomposition. So degree of decomposition just shows that as you get warmer in terms of color, that means you have more decomposition. And so what you see here is that the warmest colors are at the shallowest depths with the greatest number of fiddler crab burrows. So fiddler crabs are facilitating decomposition, but only at the shallowest depths. So that's a lot of information to unpack. And so I really sort of want to talk about, you know, the bigger picture of how fiddler crabs are influencing these ecosystems. Before going into this, I did want to mention that I didn't show any plots in terms of differences between Plum Island and Nantucket, because while there were site level differences, the two sites are unmistakably different. The patterns of change that we saw for these path diagrams, these structural equation models, were exactly the same. So the relationships were the same, even though maybe the degree to which that they occurred were different between different sites, which was a really interesting finding given the fact that the ecosystems are different. So we have crab density and initial soil strength, and we know that the combination of the two affects burrow density. Now that we have that understanding, we know that burrow density influences soil strength, which is really what's driving the rest of the system. Soil strength affects primary production. Burrow density affects decomposition, which is really influenced by soil depth. And all of these different indicators combined affects marsh area, you know, either through increases in bioturbation by fiddler crabs, changes in soil strength, just changes in primary production, potentially facilitating that. And so understanding how this crab influences marsh area is really important, given the fact that we have critical environmental change occurring in both Plum Island and Nantucket via climate change. And so here's that large map again of the Plum Island Estuary. And so here's Plum Island, and here's the city of Newburyport. And this city is right to the west of a huge barrier island, which is going to provide critical ecosystem services like buffer against stormwater, and then even aesthetic ecosystem services like birding or hunting or clamming, even just going out to the marsh and enjoying the peaceful outdoors. Meanwhile, Nantucket has very similar ecosystem functions and roles that the marsh plays. So here's a large map of Nantucket. We have Folgers Marsh, which is right up against the UMass Boston Nantucket Field Station, as well as a series of residential homes that use this ecosystem both for enjoyment, for looking out at this beautiful resource. And this marsh is going to buffer some of the effects of flooding, as well as marshes, as well as storms. So these particular habitats are going to potentially be influenced by species like this fiddler crab, which will be important moving forward. So with that, thank you so much for listening to my talk, and I'll take any questions.
Awesome. Thank you, Michael. We do have one question in the chat already. The question is, how did you survey for a range of soil strengths, and what was the variation in soil strength?
That's a great question. So essentially, we knew that from previous research that marshes north of Cape Cod were more compact relative to marshes south of Cape Cod. So we knew that from a first approximation. But I was already doing work in Plum Island. I was doing work in Nantucket, and so it seemed, because they had the different locations, the biggest extremes really in Massachusetts. You can't get any further north in Massachusetts, any further south in Massachusetts than Nantucket and Plum Island. That we saw that this was sort of a really great range of habitats to explore from. And then in terms of answering your second question, there are significant differences between the two locations in terms of sediment compaction. So in Plum Island, the sediment compaction on average is about 25 psi. It can get as high as about 60 or 70 in some locations. And in Nantucket, it's about around 10. So it's about 15 psi lower in Nantucket. That it's about, I think, if my scaling is correct, my conversion to kilopascal, which is the metric indicator of that, is about 3 or 4 kilopascal. And so, so in other words, there are significant differences, and we did find, like, really in the low marsh, like really soupy low marsh areas. In fact, I like to quote Marc on this. So he gave a really amazing marsh lecture, and in it he puts in that firm marsh and that squishy marsh. And so that squishy marsh is really, you know, Nantucket, and that firm marsh is really Plum Island. And a lot of it has to do with those differences in sediment composition from where they're getting sediment from.
Awesome, thank you. Do we have any other questions that are specifically for Michael's presentation? And if not, we can, oh, here's one from Eugene. Some benthic ecologists using cages to enclose predators have found strong flow effects due to the enclosure, often with increased sedimentation inside the enclosure. Did you observe any flow effects due to your enclosures?
So that's an excellent question. So we didn't measure flow effects, but what I can say is that we had a no cage control. So we had a plot that we measured all of the exact same things that we measured in all of the cage plots. We just put 4 flags around those in the 4 corners of exactly sort of like the same area that we would have had for the cages. And what we found is that there were no marked significant differences in terms of that control and some of the measurements that we took in for our cages. It wasn't perfect. There were some differences, but by and large, our cages seemed to be fairly representative of the community, of the system. But that is you know, a big impact. And to try to understand mechanism, it definitely will take, you know, creating larger cages, continuing to hammer home some of these questions, and you know, continuing to experiment in that way.
Great, thank you.
Yeah.
I guess at this point we can open it up to questions for either of our speakers. I see that Richard has had his hand up, I think since Marc's presentation. So Richard, if you're ready, I'm gonna patch you in here to ask your question.
You hear me?
Yes, we can.
Oh, thank you. Yeah, it's a bit of a takeoff from my colleague Jack Looney, who asked on the first speaker about non-biotic stuff. Is there a seasonal aspect to this? Having spent a lot of time on the island, it's a little different between summer and winter. Is there any impacts there? Any thoughts?
So, you know, year to year, or, you know, within year variation of how marshes function is well, you know, the short answer is no. We don't have a good idea on that. It hasn't really been quantified. Basically, like, you know, the sort of the pathways that I'm talking about haven't really been quantified. But you know, I based on, you know, there's differences in, like, for my work, you know, there's differences in the predator community throughout the course of the year, you know, both on the marine and on the bird side of things. And same thing with the resident marsh inverts, right? They recruit at different times, that sort of thing. So, you know, the short answer is, you know, the month-to-month temporal dynamics of how the marsh food web changes and what that means for ecosystem functioning is really unknown. I think that there's definitely sort of excluding the animal side of things. You know, I think that there's you know, we have a pretty good understanding of like how things like productivity and decomposition change throughout the year, right? More grass growth in the warmer months, and decomposition generally occurs in the cooler months towards the end of the growing season, that sort of thing. But you know, as for like some of the other important ecosystem functions, you know, I'm not really sure. I don't know if sedimentation rates vary throughout the course of the year, or some of the other ecosystem functions. But you know, the marsh is totally the place where these sorts of questions can be answered. I mean, you know, really you're well, you know, those of you in New England know, I mean, you know, you're limited by, like, you know, basically like how cold it is out there to get out and do your sampling, you know. So, you know, these are definitely questions that can be answered in the marsh for sure. It's not like having to go and scuba dive in 30-degree water or something like that. You know, you can go out there and sort of measure ecosystem functions at different times of the year. But you know, as for like how exactly that varies throughout the year, it's an open question.
So do we see a postdoc application, funding application for spending time down on the island in January and February?
I think it could be worth it. Yeah, definitely. If there's the funding out there, I say why not?
Next, we've got another question from Michael. How did you measure marsh area? This person wants to hear more about your assessment of crab effects on marsh area specifically.
That's a great question. So in sort of my conceptual diagram, what I meant by marsh area is more to the point of if fiddler crabs are going to have sort of localized effects on marsh structure, that potentially could scale up to the entire marsh. And so what I mean by marsh area is just sort of the amount of area that encompasses salt marshes specifically. So the amount of area of low marsh versus high marsh, and then total marsh as well. And in fact, this is a question that I've been asking for another chapter in my dissertation, which is if you do have these localized changes in fiddler crab effects on ecosystem functioning, how will that affect sort of like the totality of the marsh? And so that question is that I've been posing that towards Plum Island. Thankfully, because there's the Plum Island LTR, there is a large data set of actually going out and measuring the spatial area, the spatial extent of the salt marsh of Plum Island. And so understanding exactly more or less how much low marsh area there is and how much high marsh area there is. There have been folks that have been doing this using satellites, using transects, using on-the-ground field work to try to understand, and extrapolate from there. And then taking that and trying to use a systems dynamics model to understand how the relationships between the local and sort of the larger scale interact for marsh area. So it's just, but for the purposes of this talk, more of just thinking as sort of like a thought experiment of if you are having impacts at different scales, how will that, at different lower scales, how will that scale up to larger scales as well?
Excellent, thank you. Are there more questions from our audience? Yeah, we've got another from Eugene here. Some studies have found that fiddler crabs are getting food by grazing on benthic diatoms. What is their food in New England marshes? Is it Spartina, or diatoms? That's
an excellent question. Diatoms.
I like a short answer.
Diatoms, and some decomposing Spartina alterniflora as well, but they're really not grazing so much on the Spartina blades. They're more detritivores and benthic herbivores. So yeah, great question. Yeah, diatoms.
They're grazing on diatoms.
So there have been studies done on gut content analyses of fiddler crabs, and they have found Spartina alterniflora in their gut, but by and large, a lot of their gut is composed of diatoms. So yeah, so it's I haven't done any. I did some gut content analysis on shrimp, which was its own endeavor, and I don't know that I would want to do it on fiddler crabs. That's probably a test in patience, I'd say.
Michael, do you think that that's something that maybe not? Well, do you think that's something that varies throughout the year? Like, because there's more, dead Spartina on the ground, later in the season. I don't know how benthic diatoms change throughout the year. Probably peak productivity in the summertime. Do you think that? So yeah, is there any evidence or any knowledge of like basically like fiddler crab diet kind of changing throughout the year?
A good question. Yeah, I actually don't know if so when this study was done on their gut content, I don't know if there have been other studies done in sort of like temporal changes in their gut, or like what time of year this study was taking place. But I mean, absolutely. I mean, we know that diets change all the time with a bunch of different species. So it would make sense that fiddler crabs would do the same. Yeah, I wonder, I actually don't know a lot about the, basic life history of diatoms in terms of like what fosters, like temperature regimes foster, like good diatoms, that maybe there actually would be, their peak is like later in the season because it's colder, or maybe if it's warmer, that's better for diatoms. But I could imagine that they would probably switch to like a decomposing alterniflora marsh, which actually would, that's interesting. I wonder if that switch in diet switches sort of their ability to sort of not just change decomposition, but nutrient reuptake as sort of like seeding for the next season. Which I wonder if anybody's looked at that. I don't know. Speaking of postdocs.
Yeah, right. Yeah. No, I mean, I think that this is a, yeah, I mean, I think this is an important thing. That's why I thought this question about seasonality was so interesting, because I mean, it's, you know, it's really striking in New England marshes where there is such a big change throughout the season for sure. But like, you know, if you compare the dead of winter and you know, peak productivity in the summer, whereas, you know, in southeastern marshes, like where I am right now, it's most of that, like, you know, that brown stuff sort of stays standing throughout the year or throughout the winter. So yeah, you know, you'd think, like, you know, sort of late season, like decomposing is in New England marshes probably really important for how things kick off in the early spring. And yeah, I'm not sure if that those sort of dynamics have been investigated, but
yeah, I wonder also if like if they have like a cyclical pattern where like early season they eat decomposed alterniflora, then they switch to diatoms, then late season they eat decaying alterniflora, because maybe there's not as many diatoms in you know the early season because it hasn't gotten quite warm enough, and you know, or then again in early season there's sort of like that pulse of nutrients, you know, that you know may foster a huge bloom of diatoms in the early season. So,
yeah,
that might be it as well. So
yeah, I think that snails, I don't know about the Literaria litteria, but I know the Literaria irorata, the big snail down here, definitely switches from eating diatoms on the sediment to decomposing standing dead tissue to sometimes even eating live tissue throughout the year. So yeah, I don't know if it's like a seasonal thing that happens all the time, or if it's a year to year, you know, big spring bloom, so everybody goes for the benthic algae, or what. But yeah, those sort of dynamics are untapped and really interesting because they can control a lot of marsh processes.
I agree. Yeah. So in your experience with, I know you did a lot of work with Sesarma. So are they primarily just consuming on alterniflora, or do they change their diet as well at all, or do you know?
Yeah, I'm not sure. The purple marsh crab, Sesarma reticulatum, I'm not sure if it changes its diet throughout the year as well. Actually, a friend of mine here who's in David Johnson's lab, Serena Whittingham, is studying a similar thing. She's looking at some of these dynamics here in Virginia marshes, but yeah, it's another untapped question. Yeah,
Serena's great. Yeah, I met her up when she came up with David up to Pi. So yeah, that's cool. It's very cool.
So I had written down one of the questions that we kind of kicked down the road for the panel questions. So I'm going to go ahead and address that, and then maybe Yvonne, you can ask some other panel questions after that if we still have time. Yep. But so yeah, a really cool question you know, came up about the blue crab. You know, this is the blue crab's you know, an iconic coastal you know, East Coast species, and you know, after we heard this story about the fiddler crab moving up the coast, you know, it seems like there's going to be you know, a similar story with the blue crab, right, Michael? So like the you know, sightings are very much like that cool map that you have with the dates of fiddler crab going up. You know, it's similar stuff with blue crabs, right?
Yeah, I mean, that seems to be the case. I mean, there were before. I mean, it was slightly different from fiddlers because there'd be patchy instances that they would sort of show up, then they'd be gone for like 10 years and they'd come back.
Blue crabs?
For the blue crabs, yeah, okay. From my understanding. But but lately, like the past like 3 or 4 years, it's been more regular. It's been like on 1 year, off 1 year, on 1 year. And then I think even last year one was found, and then now this year there have been 5. So it's increasing in frequency. So it seems to be tracking that more regularly.
And so, you know, this is a really interesting tie-in between both Michael and I's research, because, you know, blue crabs, basically wherever they are in any ecosystem, but especially salt marshes, you know, they are more or less, you know, the top predator. you know, besides like, you know, ambient predators that come through like sharks and stuff like that, you know, like as for the species that are like coming in and out of the marsh, you know, every single day, blue crabs are right at the top, right? They are at the top of an important trophic cascade here in the southeast, where you know, if their numbers go down, their prey, a snail, can overgraze marsh grass. Another interesting thing that blue crabs do in the mid-Atlantic is they outcompete green crabs. They eat green crabs, and they keep green crab numbers down. And right, this is obviously, you know, a big topic of interest in New England and you know, marsh ecology in general along the East Coast is green crab stuff. And you know, the research that I've read is that if there's blue crabs around, there at least aren't going to be as many green crabs in the marsh. They might go somewhere else or something like that. But you know, so these sort of like it's a really interesting thing as blue crabs, you know, this novel, you know, important, like really powerful predator that eats a lot of different things, you know, is voracious. I once had one that ate 200 snails in the course of a 6-hour tidal cycle down here. You know, basically like, you know, talk about fiddler crab, which is an ecosystem engineer, and a blue crab, which is this, you know, a keystone predator. These are like, as these species move up, this is going to potentially fundamentally change, you know, a lot of salt marsh ecology. That was just kind of a long statement. Not really. Michael, what do you think of that?
I think that was great, Dr. Hensel. Yeah, I mean, I think about it sort of from the perspective that keystone predators, a lot of them are increasing the diversity of whatever ecosystem that they're in, that they exist in. And as you mentioned in your talk, Marc, I mean, predator abundance and richness has a really critical role in multifunctionality and just sort of functional processes, generally speaking. So it'll be interesting to see how, you know, the confluence of, you know, species range expansions, you know, sea level rise, all of these different environmental changes are going to structure salt marshes that, you know, maybe salt marshes will be just fine. Maybe, you know, they'll persist still for a long time, but it'll be dependent on sort of this like restructuring of webs, restructuring of ecosystem functioning, and maybe marshes will look different, but they'll still be a marsh. And so it'll be, I don't know, it'll be really interesting to see how that plays out. And I also have been thinking about how, you know, blue crabs, I mean, they are just, you know, they eat everything, as you said. And so they also potentially could be a control on fiddler crabs because, you know, they do also eat fiddlers in their historic range.
Yeah.
So I sort of, you know, I started this work thinking about it from like an invasions ecology perspective. But the more that I think about it, you know, they may behave like invasive species. But because, you know, I think that they probably will just behave like range expanders. They'll just be different. They'll just have their own, because they're somewhat similar to, they'll be encountering similar ecosystems to, you know, what they have experienced, but still different, you know, still not quite what they've experienced before. And then similarly on the other side, the abiotic side is like those ecosystems are similar to the one south, but maybe haven't experienced these species. So it'll just be different, I suppose.
Mm-hmm.
Not good or bad, just different.
We do, if we've got time for one more, Jack Looney's got his hand up, so we'll patch Jack in here one more time.
Just want to say thank you, guys. You refreshed my investigative powers that began in Plum Island in the late 1950s. Glad to see that folks are still involved in looking at the estuaries. And Plum Island is totally different than Folger's Creek in terms of its morphology. It lies athwart the Parker River, which maybe could have some difference in terms of your research. But thanks again, folks. I really enjoyed this evening.
Thanks, John. Really appreciate it. Yeah, that's amazing. Doing work in the, you know, in the 50s in Plum Island. Yeah, that's incredible. That's really cool.
Well, it's definitely one of the things that I've enjoyed doing research in New England is, yeah, I mean, basically the history. You know, I did my master's in undergraduate research on Sapelo Island, where a lot of the the forefathers of ecology got their start, and then to be able to do work up in Plum Island as well. In New England, where basically like same sort of thing. Some of these fundamental tenets of ecology, were discovered in these marshes. And yeah, the thing that's cool about marshes is they're, once you get past the heat and the mud and the bugs, they're really easy to work in, and you can ask really creative questions. And a lot of that is because a lot of the basic questions that were answered a while ago. And what's cool too is that things are changing, which means that the effects of certain species 40 years ago might not be the same as it is now. And as Michael and I have been talking about, the food webs potentially are changing as well. And yeah, so I mean that sort of the level of I guess the marshes are cool from an ecological standpoint because there's a lot of fun things to do, and then that is like expanded 10, 100 times when you think about their importance too, as a coastal ecosystem, as a sequester of carbon, and habitat for fisheries, and all of this. So yeah, I think as time goes on and as human impacts increase, marshes are more and more important over time.
Yeah, and I just wanted to say that, so I actually started working in marine science in the Rocky Intertidal. I did not start in salt marshes. And so it's actually working with Marc early on in Plum Island that I learned a ton. So I learned a ton from Marc, including the pitch that he just gave, which is you can answer a ton of great ecological questions in a really beautiful place. And so, and I wholeheartedly agree with that, now having worked in the marsh since 2016, and it's really just a beautiful dynamic place. And I just hope that the research continues, and it looks like it is that there are a lot of people who have a vested interest in maintaining this resource.
Yeah, I think one thing I wanted to touch on too, kind of related to that, Michael, something that I learned and really gathered an appreciation for on my time on the island was birding, basically, right? I hadn't done a whole lot of birding at all in my marsh research, and this was obviously ended up being a very big part of my project. Yeah, I mean, it worked out great that I was able to like find that birds have this effect on marsh organisms, and they have an effect on ecosystem functioning that's unique, that has not been found or described in salt marshes. And it's really cool that I was able to even find that ecological response, because I mean, I definitely got my appreciation for birds out on Nantucket. I learned most of my birds there, like from Edie and from Neil. I see Neil's on this call. I know that I'm pretty sure that I found the highest diversity and abundance of birds in Nantucket marshes because Neil was the one doing those surveys. So it is a potential little bit of a skew in my data there, because his expert eyes were not at all the different marshes. But anyway, yeah, I think that touches on basically what we're saying. And birds are such a cool example of ecologically interesting and important, and valuable aesthetically, and get a lot of people out appreciating these estuaries.
Wholeheartedly agree.
Mm-hmm.
But I had no further questions. Just wanted to say that my professor was Wes Tiffany Sr., who some of you may have heard of from the time on Nantucket. Thanks again.
Thank you, Jack. That is really interesting. And one more comment from Richard Gelke.
I just wanted to echo Jack, mostly about how old he is, but also to thank you, Yvonne, for putting this on and publicizing the field station. A lot more of this would be terrific. So thank you very much.
It is my pleasure to do this. It's really interesting. I could keep talking, and could ask a lot of questions, but I won't do that because it's getting a little bit late. But for everybody who's still with us and who is here, we will have more of these. And I thank you, Mark and Michael and Leo for assisting so much. And thank you for everyone who tuned into this and asked questions. It was really interesting. And you just want to hear more and do more. And hopefully it sparked some people's plans for experimentation in projects and grants that we can continue work down here, and maybe continue this conversation with another opportunity to have maybe just a panel where we could really get into some of these questions. I would have asked you about application to sea level rise and accretion rates, so we'll save that for another time. But this is great. Thank you so much, and thanks everyone. And if our speakers would like a final word, please feel free. But I'm signing off. Thanks everybody.
Yeah, just thanks a lot. I really enjoyed it.
Yeah, it was great. Thank you so much, Yvonne. Thanks, Mark and Leo. Thanks everyone. This is great.
Have a good night, everyone. Thank you.